Hello, my lovely anatomists and physiologists. We are ready to talk about our secondary digestive system organs, or we can call them our accessory organs. Remember, this also includes the salivary glands, which we mentioned when we talked about the mouth. You're going to be digging into the anatomy of all of these structures as part of your lab work. So I'm going to allow you this space to do that there. And I just want to fill in a little bit of physiological details, but I do want to make sure we're taking a look at these photographs of some of our lab models. So let's begin by looking at the model that's showing a cut of the liver. So you're looking here at the liver. Notice you have attached on the underneath side the gallbladder. So there's a nice anatomical and physiological relationship between the two. And notice that you have this bile duct that's emptying into the duodenum. And you see gallbladder empties into the bile duct, but also you have hepatic ducts emptying into that as well. And you can actually see right here the pancreas, and the pancreas has a pancreatic duct, which is also emptying directly into the duodenum. So these are examples of exocrine glands because they're going to be depositing directly from one duct into that next organ. So we can label pancreatic duct, and then we can label bile duct. With this other image that I have for you, this is again a model from our lab, and I did include this one as well because it's really highlighting the pancreas and the pancreatic duct, and you can see how that's emptying directly into the duodenum, and so we're getting that relationship here as well. When we talk about our liver, when we talk about the liver, we'll see its job is to produce bile. It's going to process and store nutrients. And by nutrients, remember we mean carbohydrates, we mean lipids, and we mean amino acids. In this case, I should say we mean nucleic acids, carbohydrates, and proteins. We'll see also we're going to store vitamins. So we'll see storage of vitamins A, D, E, K, and B12 here. We're going to store iron. So we have produce bile, we have process and store nutrients. Nutrients again, because I think I've messed that up. Nucleic acid, carbohydrate, amino acids, which are the building blocks of proteins. We're storing vitamins. We're storing iron. We're also going to absorb drugs and toxins. We're also going to be involved in what we can describe as hematological regulation. And I think the big takeaway here, hematological, that's talking about regulating the blood. And let's think about the regulation of osmolarity here because we are producing plasma proteins, namely albumin. And we also see it as a blood reservoir. And let me say one other little piece about the liver. I'm really filling in my gaps on my screen here. The liver is involved in breaking down proteins. And as a result of that, we get ammonia, which is very toxic. If you have a buildup of ammonia in your blood, you can go into coma, and then this can kill you. And so we see that the liver is important. A healthy liver is helping to break down that protein into the ammonia, but then also converting that to urea, which we then can get rid of at the kidney in our urine. So we'll see that. So this is the liver. We talk about the gallbladder. I'm going to switch to green color here. The gallbladder is going to store and concentrate bile. And so once we've concentrated the bile, we're going to get what we call bile salts. And the bile salts are going to be important in emulsifying lipids. A lipid, remember, is going to be nonpolar. And we're going to have a hard time breaking it down. So what we'll see is that a bile salt emulsifies it by having a hydrophilic part and a hydrophobic part. And they're going to look a little bit like a phospholipid. And so the bile salt comes in and it helps to break that lipid into a smaller droplet. And it helps that lipid then move through the solution a little bit. So when we're emulsifying the lipids, we're making it a smaller droplet, and we're also allowing it to be suspended in that polar solvent. We will see that the gallbladder production is going to be controlled by the hormone cholecystokinin, which we can abbreviate as CCK. When we look at our pancreas, we're going to talk about the pancreas as providing a buffer for the acidic chyme. We talked about that buffer as being bicarbonate rich. That's going to help increase the pH in the small intestine. We see we're going to get enzymes that are important in digesting lipids. Actually, I'm going to change this. I'm going to write digestive enzymes. So we're going to get pancreatic lipase released into the duodenum to help break down the lipids. So this is in addition to the the lingual lipase that we had. We're going to get pancreatic amylase, which is going to help break down starch. Remember, you made salivary amylase in the mouth, but it got denatured in the stomach. So now that we're back to a higher pH in the duodenum, we can release a new amylase, a pancreatic amylase. We're also going to get the release of nucleases. So remember, your ASE ending tells you it's an enzyme. So nuclease is going to help break down nucleic acids. And then we can also get the release of inactive, I'm going to say proteolytic enzymes. These are listed out in detail in your text. We're not going to list them out in detail. We're going to collectively talk about them as proteases. So that means enzyme that breaks down a protein, or proteolytic enzyme, which is another way of saying an enzyme that breaks down a protein. And then they'll be activated at the brush border, which remember is in the small intestine. So when we started with the small intestine, we said it's the star of the show. And we said, but it also needs the help of like these accessory organs. And so notice that all of these are emptying into the duodenum, so assisting with that. When we talk about the pancreas, let's also remember here we're looking at the exocrine function, but we've already talked about it in the endocrine system as having those alpha cells, which are releasing glucagon. And then we've talked about the beta cells as releasing insulin. Both of these are important in the regulation of your blood glucose levels. So make sure you're reviewing those notes that you took way back in the endocrine system and that you're current on that. Your glucagon, remember, is released when your glucose level is too low. And insulin, we've already talked about in this video series as being released when you're eating your carbohydrate and your glucose level is high. Up next, we're going to compare the difference between the absorptive and the post-absorptive state. So stay tuned. And as always, take care of yourselves and each other.